Hello, my name is Shane Dunn. This is the central nervous system presentation for my Bio 515 class. If you're one of my students watching this video, it may be helpful. I'm going to re-record this video later on in more detail, more aligned to GCU undergrad class. So first off, we're talking about neuroglial cells. Neuroglial cells are support cells in the nervous system. This means they support the neurons. We want to differentiate between the peripheral nervous system and the central nervous system. Since this presentation is only on the central nervous system, we are only going to focus on the neuroglial cells in the central nervous system. So let's first talk about astrocytes, these guys right here. Astro kind of means like stars, they kind of look like star shapes. Their main purpose is to maintain and make up the blood-brain barrier. We'll discuss this in the next slide. They also help feed other neuron cells, so other neuroglial cells and other neurons. Up next, oligodendrocytes are these guys. These guys' main purpose is to make myelin. Myelin is the insulation around neurons, and this allows them to fire more quickly. These oligodendrocytes only are found in the CNS. Microglial cells are inside the blood-brain barrier, inside the brain in the spinal cord, the CNS. These are going to act like macrophages. These are going to eat any type of dead tissue or infections inside. There are not a lot of them, but white blood cells cannot cross the blood-brain barrier, so these are the line of defense that protect us from infections in our brain. Up last is the ependymal cells. These guys will help make ventricles and produce this thing called CSF. CSF stands for cerebral spinal fluid. This is the fluid your brain sits inside. Because your brain has no contact with blood, it only has contacts with cerebral spinal fluid. This stuff, the best way to think about it, is like Sprite. It is just straight sugary water that has lots of glucose, lots of ions your brain needs to grow and function. So here's the blood-brain barrier. It is a physical and chemical barrier. So as you see, we have some blood right here, and the blood cannot get through because of these tight junctions, mainly because of the capillary outside wall, the endothelial cells of the capillaries, and the astrocytes. They make this tight junction. There are receptors that allow openings for sometimes large molecules, proteins that come through, or fatty acids. But most time, it is for only small molecules like sodium, potassium, and such. Glucose is one of those large molecules that needs to be let in because it cannot easily pass through tight junctions. We'll talk about meninges. Meninges is the layering of the CNS, so, the central nervous system. It's talking about the brain and the spinal cord. Meninges work for the brain and the spinal cord, but when we talk about it in class, we're going to talk about meninges in context of the brain. So this is a wrapping that wraps around the brain. If you've ever seen a brain from a cadaver, there's this big thick layer we usually remove, and you can kind of see. That thick layer is called the dura mater, which is where it basically touches the skull. Dura mater means tough mother. It's the thickest membrane, and it's mainly used for protection. Not much is going on. Right below that is the arachnoid mater, or sometimes referred to as arachnoid. I refer to arachnoid mother because it means spidery mother. This area helps make the ventricles, and it helps with circulation of CSF, it draining into the blood, and it moving throughout the entire central nervous system. And lastly, we have the pia mater. This stuff is like saran wrap. It actually physically touches the brain. And it's a very thin layer. It's very, very shiny, like saran wrap. It's easily punchable, and this helps maintain the blood-brain barrier. As you can see, that green line right here is the pia mater. It sits above the brain, but below capillaries and venous things. There you go. So major brain regions. So I'm not going to talk about the lobes and such. We're talking about the cerebrum, which is the front part of the brain. Actually, no, that's the frontal lobe. Where is my mouse? Sorry about that. The cerebrum is going to be both the left and right hemisphere. It is the entire top part of the brain. It is us. This is the most formed part of the brain. It is the left and right hemisphere, the occipital lobe, the parietal lobe, the temporal lobe, and the frontal lobe. This is what makes us higher functioning primates and such. Touch, vision, hearing, speech, reasoning, all this is that. The cerebellum is, I'll do a different color for us. I'll do purple. Is going to be this area right around here. Kind of looks like a wrinkly ball of some kind. And that's going to help with maintain balance and crude muscle control, or muscle memory. And lastly, we'll have the brainstem, which is going to be the lowest part of the brain. And this is going to do all things that keep us alive. So as it relates to everything else, but body temperature, sleep cycle. Some teachers may refer to this as the brainstem as the reptilian brain, and we get more and more complex as we go further up. The primary motor cortex. So the brain is broken down into motor and sensory. So when I say the word motor, it means movement. So all movement is generated in the brain. The primary motor cortex is still part of the frontal lobe. It's right here where I kind of do this yellow. It's in front of the central sulcus, the longitudinal line that breaks the brain from the frontal lobe to the parietal lobe. The primary motor cortex is where all motor things are generated, and then they go down the spinal cord and leave off where they need to go. So all fine movement starts off here. When we have fine movement, this is all movement we can control. Organ movement's a bit different. Up next, we have the sensory motor cortex, which is right behind the central sulcus and behind the motor. And what happens here is the sensations happen in our hands or feet and then travel up the spinal cord into the primary sensory area. So the biggest thing takeaway is motor goes down, sensory goes up, kind of like a freeway or a highway where there's two lanes going different directions and they can never really cross each other. Because if they do, signals get mixed up and bad things happen. So motor goes down, starts in the brain and goes down, sensory goes up. An interesting side effect of this is our brain has actually no sensation of pain. If you were to do brain surgery on someone, you'd only need anesthesia for the skin, the skull, and parts of the meninges. But the actual brain itself feels no pain. You can poke and prod it and shock it, and it feels nothing because it doesn't have sensory in there. And because of this, you can't move your brain. Motor generates in it, but there's no motor goes out from the brain. Okay, what is this weird freaky dude? This guy right here is referred to as a homunculi. It's a weird thing that we like to show students because it makes it interesting. A homunculi is a visualization of sensory and motor things. So most of our brain and nerves are going to the bigger parts of this guy. As you see, his hands are huge. Most of our brain for motor and sensory are focused in our hands, moving in sensation with our hands. That's how we grab, write, and do other things with our hands. Then you have your lips. Your lips are very sensitive. Your tongue for taste. Your ears are big. Your nose are big. Your eyes are big. But look at like your biceps and your forearm. They're super small. Not a lot of nerves are there to feel things and move things. This is another visualization just on the brain itself, and you can kind of map the guy out. So blue is going to be sensory, and then red's going to be motor. So if you were, for example, to take an electrode and poke this side, you would cause the neck to move. If you were to poke this side, you would cause the tongue to move. So knowing all this, we can kind of actually see how reading and speaking would work. So for example, popcorn reading happened in grade school. You would see a word and have to read. Well, first off, you would have to see your eyes. You have to read the word. Your eyes would have to see the light. We'll talk about that later on. As the light hits your visual cortex, called the occipital lobe. From that, it has to transfer to this area called the Wernicke area, which will process understanding of speech. So you have to look at the words, go, oh, I understand that this word right here is pathway. Okay, so I saw it. I understood what I saw. Wernicke area kicked in saying, oh, that's pathway. I understand that. Then it goes to Brocae, which means, oh, I can actually know how to move my mouth in the motor cortex to convey those words. Speaking, hearing it, you have to go from auditory to Wernicke to Brocae to motor, but you always go from Wernicke to Brocae because Wernicke is understanding speech. Brocae is actually conveying the speech. And interesting enough, we can have strokes, which is a blood loss to these areas. And if you have blood loss to Wernicke or Broca's, you can do this thing called apophagia, which is basically language problems. If you have a stroke and cause damage to the Wernicke area, this understanding of speech goes away, and you basically start producing quote unquote word salad, where I could be having a conversation and I'll say pineapple, airplane, cougar, vulture, right? And you have no idea what I'm talking about, but I understand exactly what I'm saying. I understand the conversation, but it's the words just not make sense to me. Like, I'm hearing this, you're saying that. The stroke in the Broca area occurs. I will talk like this. You talk very, very slow. Words are hard to get to. All the words are struggling to come out, type of thing. We have this concept of cranial nerves. I'm going to go over really quick the cranial nerves because this concept is usually in a tire lesson in itself at Grand Canyon University. We have 12 cranial nerves. Each one focuses on a different thing. First off, olfactory, which basically means nose, is your smell. Optical, or optic, is sight. Ocular motor is moving your eye. Trochlear is also moving your eye. Trigeminal is all of face sensation. Abductus is also eye movement. You notice your eyes have a lot of nerves because eye movement is very, very important for visual creatures. Facial is how you move and how you salivate. Vestibulocochlear, where we talk about hearing and balance in the ear. Glossopharyngeal is taste and swallow. Vagus is very, very big in the parasympathetic function. This controls all the rest and digest functions. Accessory allows us to move our head, like a yes and no, not our head. And hypoglossal helps move our tongue. There's acronyms and such that we'll convey once we get to that lesson. And up next, lastly, we're talking about daratones. And this concept basically is saying is if I were to poke like right at your hip, that would affect the lumbar 1 spine. So lumbar 1 spine right here would affect all of this area, sensory and motor. L2, L3, L4. You notice how it's very, very straight on, kind of going in the front, and as it's legs, it's more longer because the legs are usually longer in people. But those are dermatomes. Basically, sections of the skin are focused to one part of the spine. And this is how we can easily convey a bug crawling on you, is that, oh, I know exactly where it's at because only that region of the spine is freaking out. It makes moving and sensing things very easily. Here are the references that I did use. Thank you once again. Have a great day.